Welcome to the biotechnology program at Fleming College. In our biotechnology labs, the students build the practical laboratory skills needed for careers in biotechnology, pharmaceuticals, research, and quality assurance. Let's take a look inside the spaces where hands-on learning happens every day.
All right, so we saw the kind of more chemistry-centric lab. Now we're going to go over to the other side of the hall where we have kind of our more biocentric lab. This one is our tap. So we're going to come in here. Types of courses you would be doing in here are going to be your bio courses. Sometimes molecular biology and microbiology take place in here. You'll also have biochemistry, pharmacology, toxicology. So we have more of a biology focus on this side. Right now we have some microscopes set up with various slides. We have Candace over there loading a gel, and then we have another setup at the front, so we'll take you through each one individually so we get a little more sense of the space. And maybe if you want to just take a nice view of the whole lab, because it's a bit of a different setup than what we're used to on the other side.
Right, students prepare slides of B. megatherium and do simple staining on them to visualize bacteria. They also, in the Bio 1 and Bio 2 courses, analyze cell structures and look at things like bacteria, yeast, and blood, and typical plant cells. And they do this through either using prepared slides, or they will learn to prepare slides themselves and stain them with a variety of techniques.
So right now we have Candace, who is expertly loading a agarose gel. Right now she's loading various dye colors. So these dyes will either be positively charged or negatively charged. And then when we connect the lid, I can show you the lid. This lid goes on top. It has two leads, and those leads will get connected to the power supply. We turn it on, and then a current runs through the gel. And the way we have it set up, because we're predominantly working with DNA, and so DNA samples are what you're going to load into the gel. DNA is negatively charged, so it's going to run away from the negative charge and towards the positive charge. And this allows the DNA to move and separate on the gel so that we can visualize it and determine some features about its quality. And then we know how to move ahead with it. We have lots of options for loading, lots of rows we can load in. So right now Candace is loading in the first row of the gel. You maybe want to load in the second row, Candace. We can see where that second row is. So having more than one row allows us to load multiple samples. And when we have a lot of students and we have a lot of DNA samples, that definitely provides an opportunity to load multiple samples in one gel and have those all run. And probably the most you'll see is 3 rows in a gel. And you'll get to learn how to make these gels. You'll load them yourself, and you'll load your samples onto them in our first semester DNA course. And then you'll continue to use them every semester throughout the program. And just for context, Candace is our lab tech. And so she makes everything for the biotech program happen behind the scenes, keeps track of everything, orders stuff, does the setup, everything to make all our labs run as smoothly as they do. All right, so we are just back at our gel, and so we've had it running for what? How many minutes, Candace? About 10 minutes. So the current has been moving through the gel, and we can see, and it's really nicely obvious in the top row because of how many different dye colors that Candace has loaded. But some of the gels from where they were initially loaded have started moving in this direction, and the other dyes have started moving in this direction. And so that tells us that the ones that are moving in this direction, because this is our positive electrode down here, are negatively charged. And the ones that are moving in this direction, because this is our negative electrode up here, are positively charged. So the positive is attracted to the negative is attracted to the positive. And then we also have the opposite effect. Anything that's negative that's up here doesn't like being beside this negative, so it's pushing this way, and same thing with the other. But it's a really nice shot of how the dyes move based on their charge. So this is another example of something that you will see. The first time you'll see it is in second semester in your biology course, and then you'll be spending a lot of time with these cells in molecular biology. So we don't actually have cells in here right now, but as an example, this is the type of culture flask that you would use. It's designed a very specific way to have the neck slant a little bit. Air exchange can happen through the lid. And normally what we are growing in here are CHO cells, and that stands for Chinese hamster ovary cells. So it's an example of mammalian cell culture. You do a lot of research and experimentation with mammalian cell types. And so these mammalian cell types, when we're working with them and we're culturing them, we use something called a portable clean room, which is what we have set up here. There's a HEPA filter at the top, so that sterilizes the air that comes in, and it creates a clean space inside to ensure that our Chinese hamster ovary cells are always protected. And because we're a biosafety level 1, nothing we're working with can cause us working with it any harm. So it's the ideal setup. If we crank, we can see the air movement coming through. So when it's on high, it's really sucking the air in and kicking the air out to keep that environment as stable as possible, or sterile as possible. But it's very loud when we do that. So when we don't have to have it running, that's what we have. And so you'll learn how to work in here. You'll learn growing cells, culturing cells, and also setting up the cells for various types of experiments. And you'll perform an experiment yourself. So I'm gonna bring you over here, slightly different microscope setup. So this is an inverted light microscope. So the viewing is actually looking from the bottom. The way that the Chinese hamster ovary cells, they actually adhere, physically attached to the bottom of the plate. So when we're viewing, we wanna view through the bottom. We have different filters along here that we can control the type of light and the amount of light coming through. We also have a camera that we can set up to the back to take images ourselves. We can hook it up to the screen. And what we do as part of this course is we actually, in the first half, you extract and you work with the green fluorescent protein from jellyfish that makes it glow. And so we take that and we actually transform it into our Chinese hamster ovary cells to make the Chinese hamster ovary cells glow. So I am going to just show some pictures from a previous class. So this is just the regular black and white image you're gonna get of the cells. So remember, it's taken from the bottom of the plate. And when we expose these cells to UV light at a very specific wavelength, if there is that protein present, then it will fluoresce. And so the color that it fluoresces is green. And so we can see some fluorescence, and then you can use the two images and overlay them on top of each other, and you get an idea of where that fluorescence is actually happening within the cell. And then we did a similar experiment with a slightly different fluorescent protein. And what's different about this protein is that it just fluoresces at a different color. So again, we have just our black and white, hey, this is the cells. Then we look at the image under the excited UV light. So we are getting an emission of blue instead of green. And then same thing, we can overlay those two images onto each other, and we can see within the cell where that fluorescence is actually happening. And so this is a way you can monitor proteins in cells, and some researchers use it to understand where proteins function inside a cell. Is it in the cytoplasm? Is it in the nucleus? Does it happen in the mitochondria? Those kinds of things. So it helps to give some information about where things happen inside a cell.
From advanced laboratory techniques to real-world research applications, students gain the experience employers are looking for. This is where health science comes to life at Fleming College.